All living systems are made up of L amino acids. We'll call them left-handed amino acids. And there's a mirror image of these, which are the D amino acids. We'll call those the right-handed amino acids. And it's like your right and left hand. One's a non-superimposable mirror image of the other. So all life is made up of L amino acids, our proteins and everything that uses amino acids. And they're chiral, which is why they exist in these right and left-handed mirror image forms. D amino acids were originally thought to be unnatural and useless. We now know this isn't so, and they are we're finding them to play more and more important roles in a variety of biological processes. In cancer, some D amino acids appear to be upregulated. That means in a higher concentration than you'd normally find. Three in particular, D-aspartic acid, D-serine, and D-alanine, are agonists for NMDA receptors. NMDA is an acronym for N-methyl D-aspartate. And these receptors are these are ion channels that are found upregulated in nervous tissue, the brain, and they're upregulated in cancer cells as well. Now, neurologically, D-amino acid has been found to stop severe seizures, and others are neurotransmitters, and some are agonists to neurotransmitters. And we aren't sure what the entire mechanisms are because we're just finding out that these things exist. Well, the main protein or peptide in Alzheimer's disease patients is something called beta amyloid. It has about 42 amino acid chain. And beta amyloid accumulates in the plaque between nerve cells in Alzheimer's disease patients. And beta amyloid has been found to epimerize certain amino acids, epimerize in the beta amyloid at specific locations. And it turns out these locations are mainly aspartic acid and serine. And there's no good way to analyze for such aberrations in beta amyloid until now. And we've found a number of techniques that not only detect it, but can point out exactly where in the beta amyloid chain this aberration is occurring, and if there are more aberrations in Alzheimer's patients than in normal people. Well, we found that certain amino acids are elevated in cancer cells relative to non-tumorigenic cells. And this raises the question of why. Why does this happen? And this showed that they can be used as oncometabolites. An oncometabolite is simply something that is elevated in a cancer cell or a cancer patient relative to what it is in a normal patient. So as a diagnostic test, you can go in, and if you find this oncometabolite, it's an indication that there's a cancer that needs to be examined or looked for a little more closely. So in early warning systems, things like this, it's important. Pittcon offers ease of networking with a very large number of scientists in many, many different fields. And you're all together in one location. You aren't spread out all over the city like in some larger meetings. And you can easily hear a wider variety of talks than any other meeting I've ever been to. So the number of people you can access and talk to, the number of talks you can hear, it's much easier at Pittcon than virtually any meeting worldwide.